Well, welcome back everyone to our fourth and final session, Research for Tomorrow. Whether you're here with us in the room or wherever you may be online, I hope you've had a really excellent day. It feels like it's gone by in the blink of an eye. This session is going to give us a chance to hear some different perspectives from the CF community. It's going to include some updates from leading experts in the field, in the fields of genetic therapies, physiology and pharmacology, and developing ion channel treatments for CF. But we're going to begin the session with the voices of people directly affected by CF. We have two pre-recorded videos, one from Abi, who's an adult with CF, and one from Lizzy, who's the parent of a teenager with CF. And I just want to make you aware that both videos contain personal and emotive content, including discussion about mental health issues. So we'll roll the videos now. Thank you.
I'm Abi. I'm a 31-year-old CF patient. I have IF507 and DF508, so I am only eligible for Cftr, but it hasn't really worked very well for me. I am a working mother. I have 3 children, a 6-year-old and 2 5-year-olds, and life is hectic. My CF is quite strange in some ways. For most of my life, it was almost atypical. I did all my treatment, but I didn't have any hospital admissions until I was 22. I grow a Burkholderia, which I started growing at 16, which has slowly got worse and worse and worse. And then since having the children, I've had an awful lot of IVs and a lot of problems with weight. This last year, I've had 6 courses of IVs, and I'm quite intolerant to most IVs. So I have to go into hospital nowadays to have them. And it's just a progression. However much I do my nebs, I don't seem to be able to get better. And that's quite a challenge. I started Cftr in September 2020. I have to be honest, I wasn't that big a believer in it. I believed the trial data and I thought it will help me, but I didn't think it would do a massive amount because my lung function has always been high. So I wasn't expecting a massive jump. I was just hoping for stability, less IVs and weight gain, which would then increase my stability. Unfortunately, after I'd been on it a couple of days, I had a strange episode where I lost consciousness. And I was only unconscious for a couple of moments, but when I came round, I couldn't move at all down the left-hand side of my body and my left eye was blind. It only lasted 15 minutes. My husband was at home with me. It only lasted 15 minutes, but it was I felt like I'd had a stroke. I was very, very frightened by it. And I spoke to my team and they sort of they were a bit surprised by it. They didn't really know what it was. And they thought it was just stress because the purge had been so big and I'd had a hemoptysis. And they thought I was just a bit stressed. Then 2 days later, it happened again. So it's been an up and down roller coaster, just this constant, they don't quite get it high enough. I need IVs and I have to come off. And then it's just this cycle. Obviously, as you know, there's a strong online CF community, and I have spoken to a lot of CF patients about their experiences with Cftr, combined with speaking to members of staff when I've been an inpatient about what their experience of patients on Cftr is. And there's definitely a very sliding scale. At the very top, there are these miracle people who are having huge jumps in their lung function, and really it's changed their life considerably, or almost reverse their CF. Then there's a large group of people who are doing really well on it and are having a great experience, but it's not giving them these huge jumps. It's more in line with what the trial showed. So they might still be having some IVs, but not regularly. And they do feel better in themselves, but they aren't completely healthy. And those people seem to be struggling a bit when I talk to them because they are being told they should be having a miracle and they're not having that miracle. Then there's a not insignificant group of people who take after and say, I actually genuinely don't feel any different. And some of those people were already very, very healthy, but some of them weren't. And they're sort of going, well, I take it and it probably is having a long-term good effect, but I don't really feel it. There's no difference. And then obviously there are people like me who are just having a disaster. There seems to be quite a lot of people having lower level psychological problems. Their mood is really affected. They can't sleep well. They're struggling in lots of different ways. Brain fog which I have experienced, and actually is really challenging. You know, you forget things, and you can't, you know, you should be thinking something, you can't remember what you should be thinking. And there's been a lot of that, and a lot of people unfortunately feeling very, very depressed. The two things I would like researchers to focus on, the first one definitely is just CFTR generally. We don't understand what CFTR does when it's expressed correctly in the brain or in the nervous system, and both of those really need looking into. And I really hope that, as well as looking into other modulators and things like that, that the actual cause of the problems outside of modulators is looked into. So that would be my biggest one. And secondly, I really would like there to be better ways to see how Cftr is working, you know, blood tests and things like that, different doses, separating out Cftr into, you know, to use Cftr, Cftr and ivacaftor, so that people can adjust their doses a lot more than just one pill or two pill, because it's not personalized enough. So if those two things could be done, I think that would be really helpful. Understandably, there has been quite a furore around Cftr. Obviously, with COVID, it was a difficult time for everybody, and there was this big news story, this wonder drug has come out. And following on from that has been repeated stories. And every time you read anything about CF, the first thing that you read is this miracle drug, which is great. But it does mean that it's then in the mind of every single person that I know. Obviously, everyone I know personally knows I have CF, and they are reading that this is miracle cure, almost, because that is how it's presented. That's not what it is, but it's how it's presented in the media. And they want to know why I'm still doing IVs, and they can't seem to let it go, and they just constantly talk about it. And that is the same within hospitals. Actual CF team that I have are very sensitive about it, are very good about it, and don't make a big deal about it, apart from to help me. But it's all the other staff members who are brilliant and really caring, and they're sort of saying, well, why are you here, basically? Why are you not on this drug? It's a really good drug. It would change your life. Why don't you try it? Why can't it work? And it all comes from a really, really good place. But it is absolutely exhausting, because, I mean, I was in February, and every single conversation with anybody had Cftr involved. A bit more needs to be done to sort of emphasize that it's not everybody's miracle. It can't be taken by everybody. And actually, if you can't take it, it is really difficult.
I'm Lizzie Molyneux. I am a mother of 3 children. My 17-year-old son, Isaac, has cystic fibrosis. We have 2 more lovely daughters who were born following pre-implantation genetic diagnosis testing, who obviously do not have CF, and we live in Cambridgeshire. Isaac was born in 2005. We were told quite soon after that he had 2 quite rare mutations, which were diagnosed through a blood test. In fact, he was so small they couldn't do the sweat test on him. They could never quite get enough sweat from him. And so we knew from the start that he had 2 rare mutations, one of which is a stop or nonsense mutation. And at that time, it didn't really make much difference. It was just something that the doctors would say, oh, that's a bit odd. But at that time, the main buzz in the CF world was around gene therapy, which it didn't matter at that point what mutation you had. So it wasn't until much later that we realized that his rarer mutations might make a difference. And certainly I'd read in certain research papers that he has 2 class 1 mutations and that they might result in more severe disease. Isaac has grown up with CF with, like many children, a sort of roller coaster, really, where he's had years where he's been a lot better and many years where he's suffered a lot more. We tend to talk about the years of the DIOS, for example, where he had a lot of tummy related problems, and then the years of the aspergillus, where he had the fungal infections, and the many years of Pseudomonas infection. So it's been a very up and down road. And at the moment, he's in one of those dips, and so is currently on home IV antibiotics, treating another exacerbation of his lung infections. Fortunately for us, he's a very happy, laid back kind of boy. He takes everything in his stride, and we continually feel so proud and in awe of him, really. So every treatment that Isaac has now, and the burden of treatment is immense in CF. He spends hours every day completing his nebulizers, his physio, remembering to take his tablets, putting up with sinus headaches and managing his pain. And all of the treatments only treat the symptoms of his disease. And I suppose that's one of the harder aspects of knowing that we're not eligible for any of the new CFTR modulator drugs, which I would love to feel like we're treating the underlying cause of his disease rather than just those symptoms. It feels like a constant battle against the bacteria, the sinus issues, the dios issues, the fungus, and it's just one hurdle after another. To think that we could be treating that underlying cause, and that treatment might affect all of those aspects of his disease and his well-being, would be massive to us. But every time you think, oh, we're in a rhythm here, we get this, we can manage this, then something happens that we would never have predicted. In 2017, Isaac caught not one, not two, but 3 viruses at once, and was really quite unwell. We were on the ward already having IV antibiotics. He became so breathless that we ended up in intensive care. We were there in total for about 4 weeks in hospital, and he had to learn how to walk again and was sort of wheelchair bound on oxygen. And he was only 13 years old. I mean, that shook us to our core, and it shook him. I don't think he'd ever appreciated how poorly he could become and how quickly that could happen. We so desperately need something that can get us not off this roller coaster. I'm not sure that treatments in the near future will remove all of these variables, all of these hurdles, all of these worries that we live with, but they could make a massive difference. And we so desperately need this research, and we so desperately need it now. He's so brave and so strong, so fantastic. He deserves this, and we're really hopeful that in the future that research will be able to give him some of that freedom. When Isaac was born back in 2005, it was in the days where the buzz was all around gene therapy. But we were told, even in those days, that gene therapy would only work for him if his lungs were not already so damaged that the treatment couldn't get through to the right parts of his lungs. So it always felt like a ticking time bomb. We needed a treatment urgently. We needed something that would save his precious pink lungs before they were too damaged and scarred by years of infection and inflammation. And although gene therapy is only one of our options now, and there's many other options, and it's so great to see that research, which we read about all the time and we get excited about and we're so hopeful for, but we need it now. We need it urgently, because his lungs are being damaged. They are full of infection. They are full of inflammation. We are seeing a decline in his health year on year. So the pressure is on us to keep him well for as long as possible, waiting for those treatments to come along. And we're so hopeful and we're so excited. And Isaac is hopeful and resilient and brave and strong and wonderful. And I know that we'll do everything that we can to keep him as well as we can for as long as possible, waiting for those treatments. So please carry on with the research, speed it up if you can, expedite anything you can, bear us in mind. It will make a huge difference to the lives of people with CF.
Well, firstly, I want to convey a huge thank you to Lizzy and Abi for sharing their experiences and their stories with such courage and honesty. We know that there's a lot of CF patients and families watching this online today. So if you or a loved one are experiencing any concerns or side effects from your medication, then please talk to your CF team and do use the yellow card reporting system. I want to remind everyone that the CF Trust's helpline is free and confidential to use. I'm going to read out the contact details in a moment, so grab a pen if you want to note those down. The team are available from Monday to Friday, 10 till 4, and they can be contacted via email on helpline@cysticfibrosis.org.uk or by telephone on 0300 373 1000. So thank you, very powerful. We've got 4 more speakers now, followed by the final Q&A. So without further ado, I'd like to introduce Professor David Sheppard from the University of Bristol and his presentation, the Cftr Folding and Function Strategic Research Centre.
So good afternoon. I'd like to begin by thanking Paula and Belinda for the kind invitation to talk to you today. It's an honor to do so. So the CFT, so Inika, Cedric, Isabel and myself are extremely grateful to the Cystic Fibrosis Trust for this funding for the Cftr Folding and Function Strategic Research Centre. The aim of this SRC is to understand better how Cftr folds, how it assembles and how it functions. And we want that information, the knowledge that arises from this SRC, to inform innovative new therapies for cystic fibrosis. And we hope that those therapies will benefit all individuals with cystic fibrosis. So I'm sort of a little conscious of saying this after the 2 previous videos, but let's go ahead. So Elexacaftor, Tezacaftor, Ivacaftor, or Tricaftor, or Tricaftor, or Kaftrio is a transformational therapy for most individuals with cystic fibrosis. However, not everybody with cystic fibrosis, as we've just dramatically seen, benefit from combination therapy. So therefore, there's an urgent unmet need to develop new therapies for cystic fibrosis to target the root cause of disease. And 2 such approaches are first, targeted therapies for specific CFTR variants, or bypass therapy. So bypass therapy uses other pathways for anion transport across cystic fibrosis epithelia to restore ion transport and host defense mechanisms to those epithelia. And you'll hear more about that approach in the next talk. So ultimately, we as a community, we want to make CF stand for cure found. But clearly, there is still an awful lot still to do. So the top cartoon in the left, middle left, shows the folding and assembly of wild type, the normal form of CFTR. So CFTR consists of 2 transmembrane domains, 2 nucleotide binding domains, and a domain that links MBD1 with TMD2 called the regulatory domain. So the folding of individual domains begins during synthesis, and then the assembly of those individual domains takes place both during and after synthesis. So for wild-type CFTR, those domains will slot together like pieces of a jigsaw to form a fully functional protein that is then delivered to its correct cellular location, where it functions as a regulated anion channel. By contrast, cystic fibrosis variants, they will cause misfolding of individual domains. So the bottom figure in the middle shows you what happens when that variant is located in the first nucleotide binding domain. Misfolding of MBD1 then prevents the proper assembly of the protein, and then the protein is degraded. It's not allowed to traffic to its correct location and function as a regulated chloride channel. So Professor Brackman and her colleagues has demonstrated that the folding of the first nucleotide binding domain is fundamental to the folding and assembly of CFTR. So if MBD1 doesn't fold correctly, then the transmembrane domains don't assemble correctly, and there's not a proper functional channel. So MBD1 folding is absolutely essential for CFTR to function. In other data that this SRC builds on, Cedric Govaerts and his colleagues have used nanobodies. So these are miniature engineered antibodies. They're produced by llamas and alpacas. I don't know if Cedric has a llama farm, but I'm interested to find out. So the image on the left, this shows you the structure of nucleotide binding domain 1 solved by Cedric in the presence of a nanobody called G11A. And what those studies showed you, if you look at the sort of middle image at the top, Cedric identified a new conformation of CFTR in the presence of the nanobody. And that is characterized by a part of the MBD1 called the regulatory insertion taking on something of a semi-folded conformation. And what that is distinguished by is that red arrow, and that red arrow identifies a structural component of the nucleotide binding domain called a beta sheet. In contrast, in previous structures, solved structures of MBD1, the regulatory insertion was unstructured, shown by that dotted loop. So Cedric and his colleagues had identified a new conformation of MBD1. In those and other studies, they postulated that this new conformation of MBD1 was important for CFTR function. But at the same time, that conformation is inherently unstable. And so in the presence of a cystic fibrosis mutation, then the domain, and hence the protein, is liable to unfold, leading to a misfolded protein that won't be trafficked to its correct location. So Cedric's data demonstrate that in the presence of a nanobody, you stabilize the first nucleotide binding domain. So that suggests that nanobodies might be developed as new therapies for cystic fibrosis. Thus, the hypothesis behind this strategic research center is that the conformation of nucleotide binding domain 1, particularly its regulatory insertion, determines the correct assembly, its transport to, stability at the plasma membrane, and function as a regulated chloride channel. To optimize the rescue of cystic fibrosis or CFTR variants, the CFTR Folding and Function Strategic Research Center seeks to understand how MBD1 stabilization permits CFTR maturation and hence function. So this is the team of investigators that will undertake the work. So Ineke is based at Utrecht University in the Netherlands. Cedric at ULB, I am not going to try and pronounce that. That's in Brussels. Isabel is at Sorbonne University in Paris, and myself at the University of Bristol. And we each bring different expertise to the study of CFTR. So my colleagues, don't have good enough eyesight now, are expert in functional studies of CFTR using high-resolution single-channel recording. So our studies aim to understand how CFTR works, how it goes wrong in cystic fibrosis, and how small molecules restore function to CFTR. Previously, I led the F508del Strategic Research Center. And in this SRC, the team members are a postdoctoral scientist and a PhD student, and that PhD student is shared with Isabel. Sorry, I don't have any photographs of the team members, right? Ineke has expertise in CFTR conformation from when it's newly synthesized at the ribosome to when it's fully assembled at the plasma membrane. She also has significant experience in mode of action studies on small molecules, particularly CFTR correctors. Her team member is a postdoctoral scientist. I understand that he's actually got 2 PhDs, so I'm a little sort of nervous about meeting him, right? I'm sure I'm going to get a grilling. Okay, Cedric, so as I've already introduced, has expertise in structural biology and nanobody technology. In contrast to Isabel, Ineke, and myself, he's relatively new to the cystic fibrosis field, but he always publishes in sort of very high impact journals, and his studies to date on CFTR have been published in notable journals. So I'm sure there'll be more of the same. In this SRC, his colleague is going to be a postdoctoral scientist. And then finally, Isabel. So Isabel has sort of real great expertise in molecular modeling and molecular dynamics simulation applied to membrane proteins. So she studies a whole range of membrane proteins, not just CFTR. I really don't know how she does it, but just to sort of emphasize, so molecular dynamic simulation. So this is sort of understanding how proteins change their shape. So in the case of CFTR, how the membrane spanning domains will go from a closed configuration, an inverted V, to a sort of vertical position where the membrane spanning domains are arranged vertically and allowing ions to pass through the channel. Understanding those conformational changes are very important to understanding how CFTR works, how it goes wrong in cystic fibrosis. Her team member then is a PhD student that I'm co-supervising with Isabel. So to summarize, the team members bring a variety of powerful, innovative techniques to study how the CFTR structure, how CFTR folds, how it assembles, and how it functions as a regulated chloride channel. So, to assist the development of improved therapies for cystic fibrosis, the CFTR Folding and Function Strategic Research Centre aims to understand better CFTR folding, assembly, and function to design completely new ways to rescue faulty CFTR, the root cause of cystic fibrosis. Thank you very much for your attention.
Thank you very much, David. So next I'd like to introduce Violeta Railean from the University of Lisbon, and she'll be presenting on restoring airway function in CF by stimulation of alternative chloride channels. Violeta.
Hello, everyone. I'm Violeta Railean, a PhD student from University of Lisbon, and I'm very thankful for the opportunity to present my work within SRT project entitled with Personalized Therapies for All, Restoring Airway Function in Cystic Fibrosis Using Alternative Chloride Channels. As you know, in healthy airways, we have a functional CFTR which provides a pathway for the chloride secretion, and this leads to water movement to the airway surface. And in cystic fibrosis airways, this protein is dysfunctional, so there is no water movement to the airway surface. And as a consequence, we have low hydration level and the accumulation of thick and sticky mucus, in contrast with what is happening in healthy airways, where we have a high hydration level. And in cystic fibrosis airways, the mucus is a proper environment for bacterial infection and chronic inflammation. However, in airways, we also have the presence of alternative chloride channels, which can also provide a pathway for the chloride secretion. And our hypothesis is that by activating these channels, we can increase the chloride secretion, which will lead to water movement and that will hydrate the airways. So TMEM16A, it's one of the candidates and seems to be a promising therapeutic target to restore airway function in cystic fibrosis independent of CFTR mutation. Our principal aim is to identify and functionally validate new drugs as TMEM16A modulators, more specifically TMEM16A activators. And our work comprises 4 major steps. And the primary screen was performed using drugs that are already approved. And the first chapter was realized by our collaborators from Jeffrey Bigman's lab in Netherlands, where they used 700 of drugs, and they analyzed what is happening to the nasal organoids that have 508del homozygous for CFTR. And they analyzed which compounds were able to increase the swelling of these organoids. And this when the organoid swells, it means that there is water movement to the inside of the organoid. So they found 38 compounds that were able to increase the swelling. However, they didn't know what channels were responsible for the swelling that was observed. So for that, we use these compounds in the secondary screen, but using a model which is Cftr mutation independent. So there is no Cftr expressed at all, only TMEM16A channels. And also the cells that were used, they were fluorescent. And so what's happening when we have high concentration of extracellular iodide, it flows into the cells through TMEM16A channels, leading to fluorescence decrease, or cells become more darker. So what we see, it's a curve like this. And in case when we have an inhibitor, there is no iodide flowing through this from TMEM16A channel, so there is no fluorescence decay. When we have activator, in the same period of time, this fluorescence decay is faster. So the cell becomes darker faster. And as a negative control, we used NE9, which is a specific TMEM16A inhibitor, and EACT as a positive control, which is a TMEM16A activator. And we used these 38 compounds, and we incubated cells for 25 minutes, and we found one inhibitor and one activator. When we incubated the cells with the compounds for 24 hours, we found one inhibitor, which was the same after 25 minutes, and 8 activators. So 9 compounds were shown to increase the activity of TMEM16A. As the most promising compound to be studied further, we chose which was the most potent, because even at the lower concentration, it was able to increase the activity more than our positive control. And this compound had effect after 24 hours. So first of all, we wanted to know if this compound is specific for our channel. So for that, we used 2 cells in parallel that have normal TMEM16A protein level, and also other cells that have low TMEM16A protein level. And it's possible to see that the fluorescence of the cells become slower. The cells' fluorescence decays slower, which means that these compounds are acting actually on TMEM16A specifically. So the next step, it was compound validation. So we tried different techniques to show that this compound is effective for activating TMEM16A. So first of all, we didn't know if this compound were able to increase directly TMEM16A by increasing its function, or because we have more channel numbers at the cell surface. So for that, we performed a quantitative analysis of the channels at the cell surface, and it's possible to see that after treatment, the band is darker, which means that there is more proteins at the same amount of the cells. So it seems that this compound it's increasing TMEM16A channels at the cell surface. Next, we wanted to see what is happening to the chloride transport across these cells. And we used activator of TMEM16A, which is a ATP, and which is known to activate TMEM16A. And what we normally see is that we see a response, a negative response, which it's tied to chloride transport. And after treatment of the cells with our compound, this response was lower, which was unexpected. And so further experiments are needed to understand the mechanism of action of this compound. So we are still ongoing with this chapter, and our next step is compound validation in patient-derived samples. So we are expecting to use nasal or bronchial epithelial cells, and to perform airway organoid swelling analysis in organoids that do not have Cftr expressed at all. And we want to know if this compound is able to increase the activity of TMM69, so that can lead to swelling. And another important part is to study the hydration level of this compound that this compound can provide. So we expect that after treatment with our compound, there will be a secretion of the chlorides, which will lead to water movement. So it will increase the hydration level. So the airways become more hydrated. So as a summary, from collection of 700 compounds, from primary screen we had 38 compounds resulting. After secondary screen we have 9 activators, and we choose one activator to be studied in more detail. So I would like to acknowledge all my SRC group for all the support, and also the Cystic Fibrosis Trust, that made this work to be possible. Thank you.
Well, thank you very much, Violeta. So now I'd like to introduce Professor Pietro Cicuta from the University of Cambridge, who'll be presenting on a personalised approach to mucociliary clearance. Thank you.
Thank you very much for the invitation and give me the time to present the progress in our SRC project. So the last talk introduced mucociliary clearance. At least in the lung stage, this is the big thing that can go wrong with CF. So you're now familiar with the cartoon on the top left. There's that sort of shows an idealized cross-section of a lung tissue with multiciliated cells and some goblet cells. Goblet cells are the ones that produce most of the mucus, but the majority of the epithelium in a upper airway is composed of ciliated cells. So the little black stripes at the top of the cartoon are cilia. These are specifically motile cilia. They move left and right at around between 10 and 15 times a second, and they do so in a way that drags the layer of mucus which is on top of them in a particular direction, which is upwards, so that the mucus gets then thrown into the digestive tract. That's when it works well. That's called mucociliary clearance. Now, if the mucus goes too stiff, as you heard before, that can happen because not enough water is being sort of pushed into the mucus, and it's essentially too dry and therefore stiffens. The cilia become unable to move it, and then if the mucus is not constantly kind of refreshed and moved away, bacteria can kind of grow. That's at least sort of the simple explanation why these tissues can be colonized by so many bacteria in CF. So the picture on the bottom left is an electron microscopy just showing you kind of individual multiciliated cells. So mucus has been studied for many years. That reference from 2012 was a big step forward in our understanding of mucus. Mucus is made from proteins called mucins, which form a network. Imagine spaghetti, but spaghetti that stick to each other, so perhaps British overcooked spaghetti. And it's actually a very difficult material to study in itself. I mean, you can harvest it from cells or from sputum, etc. But those mucins are very delicate. As you change salts and water contents, they do very different things, and they age. So it's been there are groups who devote themselves to studying mucus, but it's a very difficult material. So one of the things we wanted to do was bring together a consortium where we could study the mucus as much as possible in situ, I mean in lab, in vitro, on petri dishes, but in the presence of the cells, together with understanding how the cilia interact with this mucus, and also bringing in expertise on growing the right cells with the right mutations. And that's all in an effort to come up with a system where we could then sort of look at drugs on the same genetic background as particular patients. We heard earlier kind of the importance of personalized medicine and the promise of perhaps of personalized medicine, specifically in CF, where kind of the root cause of the disease is very obvious, but the genetic background and confounding factors are very complicated. So I show there that failure of mucociliary clearance happens also in other diseases. Primary ciliary dyskinesia is when the cilia themselves don't work well, and then there's a whole range of sort of inflammatory states that include asthma and COPD and chronic conditions that also sort of degenerate the lung. But CF is very specific in the fact that it's the mucus that is preventing mucociliary clearance. So our network is sort of Cambridge-centered, has a big focus in Cambridge. I myself am the physicist, so my role is to bring together the device, the imaging of the cilia, and various sort of functional assays. We work very closely with the stem cells, and iPSC stem cells are one of the kind of promising model systems. There's a connection to clinicians in Papworth, although the pandemic actually kind of prevented some of that work from happening. And then we have kind of two labs in the US. The lab in Stanford is a chemical engineering team that does work on the mucus, and I'll tell you a bit about each of these labs. And the connection at UNC is very important because University of North Carolina is one of the kind of globally leading places for all aspects of lung disease and lung science. So now briefly, I have a few slides from each of the nodes. So in Stanford, the chemical engineers have a big track record in understanding how complicated liquids flow. That's called rheology, and in developing interesting equipment to study these things. And what they're doing is devising a very miniaturized magnetic needle, less than 1 millimeter long, that can be put on petri dishes in the presence of the living cells, all at the right temperature and the right sort of fairly physiological conditions, and thereby to push back and forth this needle and measure its mechanical response, which is dampened and controlled by the mucus, and get the mucus properties in a condition that is much more physiological than if you were to sort of suck up the mucus and put it in a conventional instrument. So the picture on the right is the existing instrument. This instrument has already been working for the past 3 or 4 months, and in our kind of monthly meetings, we've shared preliminary data, but because it's still sort of unpublished, they wanted me to show just schematics. So this needle can be pushed and by a magnetic field and held, and because the mucus is elastic, it will push back, and you can get mechanical properties from that. Or you can push it left and right and measure whether, depending on the force you put in, the needle is in phase or out of phase with the force you're putting, and that again is another way to kind of extract mechanical properties. These are schematics from my own lab. If you've been following research on growing ciliated cells, then the most common technique is to grow them on a porous membrane where they receive nutrients from below and they're exposed to air from above. This is called an air-liquid interface culture. It happens in petri dishes. You can pick up this membrane, kind of fold it around some solid sheet, and then look from the side with a microscope. And if you do that, hopefully these movies can be made to play, but they're not playing. The movie on the left, maybe you can click the... Perfect, thank you. The movie on the left is pretty much in real time. It shows you the profile of these cilia moving back and forth. We collect much higher frame rate than with a normal camera, so we can actually slow it down and see the shapes of the cilia. But this I've actually showed you in previous years, maybe some of you remember. But this is a new experiment where at the same time we're doing what's happening on the right. We have a dye, which is a cage dye. We shine a very small spot of UV light that releases the fluorescence. And then this spot, we can track how it expands and how it's moved along by the cilia. Now, the spot is so small that we can actually decide where to put it within the cilia layer. And we can study, therefore, how the cilia are pushing the liquid inside the cilia layer, left or right, as a function of how far away you are from the cilia base. So this is quite new. Perhaps let's focus on the graph at the bottom right. In orange, you see how wide that spot becomes as a function of time. So we track it for 3 or 4 seconds. Because of diffusion, any sort of little tiny blob will diffuse out. But in addition to diffusion here, it's a system where there's constant motion driven by the cilia. And in blue, you see that the spot sort of initially moves very fast and then kind of moves in the same direction, but a bit slower. So we're trying to understand this. So this is actually in a first experiment where the mucus has been washed away, but the cilia are covered themselves with mucins. And the detailed sort of hydration state will be important also in the periciliary layer, even without sort of the thick mucus at the top. And we can make this more complicated as we sort of understand it better. I've spoken to this already. At UNC, so we have a very important collaborator. This is the lab of Scott Randell. They've been running essentially a cell line biobank and related experiments for many, many years. So he told me to tell you that there is this paper that they're very happy with, which is currently a preprint, where they did two things. They have a new way to grow cell lines, which they're very proud of. They start from primary cells, nasal primary cells, but they found a new way to expand them quicker and more robustly for more generations than it was previously possible. And then they use this new kind of culture system to study a new molecule that is summarized here as CC90009, which I'm just a physicist, so I can't really tell you what it does, except that it's a transcription factor that changes the activity of one particular gene. And what you see in these data, which are data of the kind of ionic permeability of tissues, is that the control, which is the gray, sort of doesn't change over time, whereas in the presence of the CC9 thing, there is a strong signal. So this, sort of in plain words, means that the compound they've found is restoring, they say, to within 20 or 30% the physiological state of the CTFR channel. So for more details, please click on the kind of open preprint. And last but not least, our collaborators at Stem Cells have learned how to kind of program the differentiation from iPSC cells, which are the cells in blood, all the way into epithelial systems. And they've done very careful kind of checking that the system is expressing the same proteins that you would expect from an epithelium, and the same cell types, so the goblet cells and sort of other minority cell types that you would expect in an epithelium. And this is super important because you can then hope to replicate kind of the tissue and knock out, create one mutation, but in the presence of the exact same kind of genetic background, and compare sort of like for like. And in the near future, perhaps we can also introduce more cell types from the same differentiation pathway. For example, have co-cultures that start to include the immune system and inflammatory states with the same genome. And this would be very complicated to do just starting from biopsies and primary cells. So this is a picture just showing you on the right with the immunostaining that's sort of through making organoids and then taking those organoids and kind of putting them down its alley, you can get ciliation and you can get the right expression of all the right proteins from this system. So this is the lab of Ludovic Vallier, and Marta is here in the audience. So briefly how it comes together. Oh, I've got red carded. So we're trying to do each what we do best, but we're tightly knitted. Post-pandemic, we've even sort of met in person and started visiting each other. And we're heading towards really mixing up our biological and biological systems and technology systems to kind of understand mucociliary clearance in a personalized way. Thank you.
Well, thank you very much, Pietro. So I'd like to introduce our final speaker of the day, Professor Stephen Hart from UCL Great Ormond Street Institute of Child Health, and his presentation, CRISPR Gene Editing as a Novel Genetic Therapy for Cystic Fibrosis. Thank you.
Okay, thank you. Afternoon, everyone, and thanks to the organisers for the invitation. So I'll move on. So I've been working in the field of gene therapy for CF for about 30 years now. In fact, this is my 30th anniversary. So that tells you how successful we've been. But to be honest, it's been an amazing journey, quite a roller coaster journey, but we're not there yet. So with this mixed record so far, why continue? So what we've found so far from previous gene therapy trials, what they've all indicated is a need for higher and more durable Cftr gene expression. So why continue? Well, there are new technologies have emerged over recent years that give us new hope for, at least for trying out new approaches, and the need is still there. So we've heard very eloquently today that the modulators are fantastic, but aren't working for everyone. So I'm going to tell you about some of the research we've been doing in our SRC, and a lot of the members are here, and if I have time, I'll introduce you to them later. And I'm going to talk about another project funded by the CF Trust on our mRNA research as well. So in this presentation, I'll describe the approaches we're taking to new CF therapies, how we deliver them, which is delivery is key to everything to get anything working. The models for testing the efficacy of these therapies, the models tell you everything about your therapy, how functional it is, and these are critical to get right as well. In vivo delivery, so that's obviously important for testing efficacy in a real lung situation. So this is our workflow for how we test our therapies. So we start off collecting primary cells from CF donors, and we have a collection of cells with different kinds of mutations. So we then culture the basal cells, which are the pluripotential cells. These have the potential to differentiate into all the different cell types in the upper airway, the ciliated cells, the mucus-producing cells, etc. But the trouble with these cells is that they lose that potential after 2 to 3 passages. So at that point, what we do in number 3, we transduce them with a lentivirus encoding a gene called BMI1. And I haven't got time to go into the details about this, but essentially this is an anti-senescence gene. It stops the cells from aging. And this gives us the potential for almost unlimited expansion of our cells. And that gives us a fantastic resource for developing and testing our therapies. And going up to the top right, you see the air-liquid interface culture. And this is how we culture the cells in a way that they differentiate to form all the cell types of the trachea, the bronchi, the bronchioles. So that's the ciliated cells, mucus-producing cells. And in particular, when we culture CF cells in these models, one of the problems we have is they produce copious amounts of thick, sticky mucus. So this is our model that we use for testing our therapies, and it's a very challenging model. So we can edit the cells with Crispr, and I'll mention a little bit more about that in a minute, either at the earlier stage, pre-ALI, or we're also testing it actually in the ALI situation. When we've finished doing our therapy with the approaches we're interested in, which are CRISPR, mRNA, and also siRNA, we can then use these ALI cultures for functional and molecular testing to see if the therapies are working. And for that, we might look at restoration of Cftr protein, the effect on the thickness of the airway surface liquid, because as we've heard, that's a particularly important property of the airway. And we can measure the restoration of chloride transport, so the ion transport function of the protein. So I don't have time to go into this, but this is the nanoparticle, the critical delivery formulation that we've developed in our laboratory. If I could have the video running, please, that would be great. It's bottom left corner. So we formulate these nanoparticles just mixing together a mixture of peptides and lipids with our RNA molecules. And if we get the mixing right, you get these very uniform nanoparticles formed. And they're jiggling around because they're being bombarded by water molecules. So this is Brownian motion. So it gives you an idea of how small they are. So the properties of these particles, they're epithelial targeted. So they target receptors on the epithelial surface. They're able to escape the endosome, which is a key step in getting transfection. We've done a lot of research on enabling them to penetrate mucus. They're resistant to nebulization, and we can package various types of RNA or indeed DNA cargoes, and they show low toxicity. So in the first approach I was talking about with messenger RNA, so this is, I'm sure everyone is familiar with the RNA COVID vaccines from Moderna and Pfizer. So this is akin to that technology. So in the top right, you see the RNA molecule encoding Cftr. So we package that into our nanoparticles, and we've transfected here the basal epithelial cells in the first instance to see that the therapy is working. And then we've analyzed the protein produced by Western blot. And in the early time course from 4 hours to 8 hours, you can see that protein starts to be produced as early as 4 hours. But by 8 hours, it's getting stronger. And interestingly, the initial band B, which is the immature form, is starting to deplete, and increasingly we're seeing relatively larger amounts of band C, which is the mature form of the Cftr protein. So here also what we've looked at is the combination of mRNA with lumacaftor, because we're interested in looking at combination therapies. And this could be particularly useful in patients with undruggable mutations at present. So it might expand the use of the Vertex drugs. When we look at the longer time points at 24 and 48 hours, we see that the protein is actually still getting stronger, particularly in 5 and 6, where we have the combination with the Vertex drug, lumacaftor. And it's still maintained at a very strong level at 48 hours, whereas the mRNA in lane 4 without the Vertex drug is starting to be depleted. So this is an angle that we're very keen to pursue, this sort of combination of therapies to get improved expression of Cftr from mRNA. So the next question is, okay, so we're getting protein made. Is it actually doing anything? Is it functional? So this work is done in collaboration with Deborah Baines at St. George's, who I don't think is here today. And this is a readout from Ussing chamber experiments. So we transfect the mRNA into our alley cultures, as you see on the top right. And then we measure, we activate the chloride channel by addition of an activator for scolin. And you can see the non-CF cells on the far right, the dark blue, give a very strong increase in the current. But this is almost matched, it's not really a significant difference, by our combination of mRNA and the lumacaftor drug. And likewise, when we then add the Cftr-specific inhibitor, although there's a bit of variability with the non-CF control, the mRNA plus VX-809 therapy is giving a very good response as well. So we're getting functionality from these transfections. So just to summarize where we are with the Cftr mRNA, so we've prepared nanoparticles and biophysically characterized them, and they've been optimized for mucus penetration and transfection in alley cultures. We can transfect basal cells and restore Cftr protein production that persists at least 48 hours. And in alley transfections, we restore ion transport And I didn't show the data, but in vivo, mRNA gives much higher level transfection than plasma. It's approximately 200-fold higher. So in the last couple of minutes, I'm going to quickly tell you about some of our work on gene editing, which we're continuing in our SRC. This is work that was started in Patrick Harrison's lab on a mutation, this mutation here, the 3849 plus 10 kb C to T. This is a particularly attractive mutation to start with, because all we have to do, it's caused by a mutation in intron, where it creates a cryptic splice site. So there's a splicing defect and produces an inactive protein. So Patrick, whoops, that's not working. Patrick, that's your slide. What it's meant to show is that with Patrick's lab designed CRISPR molecules that flank the mutation site and splice it out. So thereby removing the splice site and enabling proper splicing to occur. So in these experiments, we've shown that we get the deletion is very efficient. So we get up to 60% deletion of the target site. And then on the right, you see that we reduce the length of the mRNA from the uncorrected to the corrected by our splicing. So we're getting correct splicing again. And I'll move on to, again, the important thing is the functional testing. And again, this was done with Deborah Bain's lab. And likewise, again, we've shown activation with forskolin and blocking with the Cftr inhibitor, showing greatly improvement in functionality of the channel. And on the bottom right, you see a specific trace. So I've had my red card. So I just wanted to show you the in vivo data we've got showing that we're transfecting in vivo in the surface epithelial cells, which would be the target for the kind of therapies that we're developing either with RNA, which is in the middle row, or with Cas9 CRISPR gene editing in the top row. So swiftly moving on, I just want to thank everyone who's contributed to this project and the members of our new SRC who are highlighted in red, and all of the students, Miriam, Izzy, and Lucia, are here today, so they'll be happy to talk to you, I'm sure. So I better stop there before I get kicked off the stage. So.
Thank you very much, Stephen. So if you want to take a seat here, and I'll invite the rest of the panel up. We've got a few moments for questions. I'm going to start with questions from our online audience. I'll be going to Belinda. She's got her own handheld mic this time. And then I'll come back to the room to see if there are any questions. So Belinda, what have we got from our online community?
So the questions are still coming through.
Okay.
But we do have one question for Violeta, and it's about whether or not it's about how the medication would be delivered. Would it be delivered via a nebulizer when you go that far?
Yes, because
So we just get your mic.
it's not working. Oh, sorry. So we think that it's better to be as nebulizer because our channel of interest it's located on airway surface. So we need to activate from apical side. So it's better to be as a nebulizer.
Thank you. So while we wait for more questions to come in from online, does anyone in the room have a question for any of our speakers? Yes. So bring the mic down here. Thanks, Alex.
So with inhaled mRNA, it looks like you're getting expression to 48 hours. Is there anything you can do to increase that? Because presumably for a clinical product, the suggestion would be you have to deliver that every 48 hours with potential toxicity from the delivery particle as well.
Yes, thanks for that. So yeah, I didn't want to give the impression that it just stops after 48 hours. We just haven't got more data. We haven't extended the study yet. So looking at if you do a Western blot for CFTR in non-CF donor cells, the level of CFTR is often quite low in Westerns. We're still getting very high levels relatively with our mRNA at 48 hours. So it's certainly decaying, and that's what you expect with an mRNA. The mRNA itself, we've shown, has a half-life of about 7 hours, so that's very short. But it's the protein, and I think that might be what is happening with the lumacaftor we're adding. It might be stabilizing the protein a bit further, and so we're pushing out the stability of the protein, and we'd like to get guidance on how long or how frequently redosing would be acceptable, but at the moment we're looking for a week or so.
Great, thank you. I'm going to come back to Belinda, then I'll come to you next. So Belinda, I think, has a question come in from online?
It has. This is also for Steve. So it says, presumably any future mRNA therapy, very similar line to Violeta's question, would need to be nebulized. Any idea of how durable that might be?
Yes, so the nanoparticle is designed to package the mRNA to keep it stable, protect it from extracellular enzymes that might degrade it, and also the shearing forces that are encountered in nebulization. So the peptide, so everyone's familiar with LNPs, which are the lipid nanoparticles that were used in the vaccines, but we've got an additional component, the peptide, which really does bind very strongly to the nucleic acid and helps maintain its stability. So we've shown it can be nebulized. So yeah, that's a very important question.
We've got a question just here, and then I'll come to you next.
I think you've partly answered the question. So I was thinking of CF in different forms in terms of acute exacerbations and everything, so the rheological properties of the mucus would change as well. So is there a future collaboration between the two of you to sort of think about, how those things can be altered?
That's just what I was thinking as I was listening. And we are collaborating with a company called Algae Pharma, who make a calcium chelating agent, which makes the mucus more fluid. And in fact, I didn't have time to mention it, but the data I showed was also using their oligo reagent because, as I said, the mucus is so thick and almost impenetrable. You've got to give something to help get through that barrier. Yeah. Thank you.
Okay, thank you. So we'll go straight to another question in the room, just over here, please.
Hi, my question is for Violeta. Nice talk, by the way. I was wondering, so from your previous answer, I'm understanding that your target would be mainly the epithelial, like the airway epithelial. But I was wondering, is this channel present in other places? And we also know that CFTR is not only involved in transport of chloride. So I was wondering, is this alternative channel just chloride deputed to transport chloride, or will intervene also in the other kind of problem we see by the abolition of CFTR, like, I don't know, transport of bicarbonate or thiocyanate?
Yeah, so this channel is also present in other organs. However, it has higher expression in airways, and we think that by activating the individuals with CF that have problems with respiratory tract can benefit from this. So it's more important for the lungs. I don't know if I answered your question.
No, just wondering if it was transporting an ion beside chloride in the airway.
It's having a very little conductance of bicarbonate. So, but
Okay, thank you. So we've got a couple of minutes left. Anything in from online? Okay, so let's. Might as well take Belinda's microphone.
It's a question for David, just with the nanobodies. Maybe a delivery question, but possibly the better one to ask is that just going to work for F508del, or do you need to have different nanobodies for different mutations, or how do you see that expanding?
So Cedric has a library of antibodies, and so I would anticipate it's probably going to be different antibodies for different CFTR variants. But as you indicate, we still you know, these are experiments that still need to be done. Yeah.
Okay, thank you. Are there any final comments or questions? Okay, right at the back there. Jane.
Thanks, really good session. Question for Steve. Steve, have you tried other mucoactive or mucolytic agents, perhaps ones that are licensed, rather than maybe predicating it on something that may or may not get into the clinic?
Thanks, Jane. Yeah, so we've tried N-acetylcysteine. That doesn't work as well. It's not really our nanoparticle relies on peptides with a cyclic targeting head group. So this reducing agent isn't so compatible with our nanoparticle. And yeah, we got the best results with the oligo G, but I'd be very keen to try other mucus modifying agents. So if you've got any suggestions, happy to talk about it. Thanks.
Okay, Belinda, one more.
Again, this is for Steve. Bear with me. So the question is your RNA modified with pseudouridine? And also, has your aliculture approach been compatible with microbial infection assays?
So the mRNA is, in fact, the Cftr we got was from BioNTech. So we're using their technology that they used in their vaccine. So I don't know exactly if it's modified, but I think they are using chemically modified mRNA. And we're now making our own mRNA. So we're trying to make improvements in our lab to the mRNA. And certainly we find when we put in the chemically modified bases, the N1 methyl pseudouridine, that gives a considerable improvement to the level of expression and the stability. And the alicultures question, so I don't know anything about doing bacterial assays on alicultures, but maybe other people do. Debbie does. Yeah, we can. Yes, we can do bacterial assays. Yeah. Okay. Thank you.
So we're due to wrap up, but Narshan, I saw your hand. We've got one final question.
Yeah, this one's for David. It's a bit of a dim question, I'm afraid. Did you say your nanobodies target the NBD?
Cedric has a variety of NBDs, a variety of nanobodies, and so some of them are targeting the nucleotide binding domain. So clearly they need to be delivered into cells.
Yeah, how does that happen? That was the question.
Yeah. So that's going to be part of the challenge of developing nanobodies as a therapy. Yeah.
Great. Well, thank you very much to our panel. So we're going to start wrapping up now, so I'll invite you to return to your seats. While I'm doing some final housekeeping notices, I'm wondering, should we bring the lectern back up? Or are we going to are you okay with the handheld, Lucy? I think we'd rather have the lectern, and then we get the branding at the front. So just while we're sorting that out, a couple of things I want to mention. The Trust will be in touch with everybody. Make sure I'm in the camera. The Trust will be in touch with everybody with information on how you can re-watch all of the sessions and also a feedback survey. So please do take part in that and let us know what you thought of the day. We recognise that there were some questions from the earlier sessions that we didn't have an opportunity to address. So we'll look at how we might be able to feed those answers back to all of you via the online platform. And for those of you in the room, when you leave, please don't go home in your lanyards, as lovely as they look. We want to be able to reuse those. So I'm going to hand over to Lucy in a moment. I just wanted to say on a personal note, this is my second year of facilitating this conference. And, you know, as you all know, I'm not a clinical researcher. I am absolutely not an expert by experience. I definitely know less about CF, and a little bit more now after today, but I know less than anybody else that's here in this room or watching today. And yet, once again, I find myself leaving your conference feeling so inspired and moved and impressed by the passion, the commitment, the dedication, and the energy of this community. So thank you very much for having me. It's been a real privilege to have this insight into the work that you do and the experiences that you've had. And I'm now going to hand over to Lucy Allen to formally draw things to a close.
A huge thank you to the Outlook Group, who have been working incredibly hard in the lead up to the event and during the event. So thank you, Ali, Laurie, there's nothing we've thrown at them yet that has made them look anywhere near flustered. So thank you. You've been a pleasure to work with. I want to say thank you to our Trust Home team. I'm not going to be able to mention you all, but I just want to give some personal shout outs and thank yous to Belinda, Nicola, Paula, Jacqueline and Danny for all the hard work that you've put into today. I know it's been a lot of work, and the agenda from my perspective has been absolutely wonderful, so thank you for allowing that to happen and bringing it all together. I also want to say a heartfelt thank you to Abi and to Lizzy for sharing their personal stories with us. They were absolutely fantastic and obviously give us some context and very powerful things to think about for the future of the research going forwards. As Catherine has said, the conference site will be open for a month, hopefully from this Friday, and the Trust team will be in contact with you to let you know how to access those recordings of all of the sessions, including some of the breakout sessions that you may not been able to attend. There will also be a feedback questionnaire. Please do give us your feedback. The feedback from last year was incredibly helpful to us, and also we want to improve this for those in the room and for those joining us remotely going forwards. I also want to say a huge thank you to Catherine. She is fabulous. You've been fabulous again. It's been a pleasure working with you. Thank you for today. And I think I have a couple of notices for the diary. No? No? I had some slides? No? Okay, so I just want to remind you that Wear Yellow Day is coming up, which is on Friday the 17th of June, which will be the culmination of CF Week. So please do get involved in that from a fundraising perspective and also just supporting the community. To save the dates, so we have the annual clinical trial conference next year on the 3rd of March, which will be hosted in Cardiff in Wales at the International Convention Centre, and the next UK CFC conference in 2023 will be held in September. We've decided to move the date because it allows us to go back to the original format we had before we moved to earlier in the year, where we can include some of our students that we fund over the summer period. So we think it's really important to include those really early stage career researchers in our day as well. So hopefully we'll let you know the date when we confirm that. And finally, please do keep an eye out for the second stage of the research priority refresh, which will be opening hopefully in the next couple of weeks. And we really do want all of your input, particularly from the research community as well as the CF community and the clinical community. So thank you again. My pleasure to close the conference. I wish you all a lovely afternoon, evening, and hope to see you next year. Thank you.